Welcome to The Explainer. Today, we're tracing an absolutely incredible scientific journey. We're going to follow exactly how researchers took 18 patient samples to unmask a really complex, hidden skin cancer using some cutting-edge transcriptomics. We'll be breaking down a fascinating recent study from Nature Immunology, and we're taking a true A to Z approach here. We'll follow this rare disease all the way from the physical tissue biopsies in the clinic straight through to the complex computational code that finally unlocked its secrets.
So, you might be wondering, what makes this disease so tricky? Well, early-stage cutaneous T-cell lymphoma, or CTCL, is quite literally a master of disguise. It masquerades as benign everyday conditions like eczema or psoriasis. Clinicians will look at a patient's skin and just see dry, itchy patches that look exactly like regular inflammation. Because of this, the disease is incredibly difficult to nail down before it progresses to a fatal stage, meaning patients often face a terrifying delay in getting the right treatment.
Here's our roadmap for today. We'll start with the great imitator, then dive into the prep of those 18 patient samples, break down the data processing pipeline, explore the resulting cellular atlas, and finally interpret the unexpected B-cell link. Let's jump into Section 1, the great imitator, diagnosing cutaneous T-cell lymphoma.
Because CTCL hides so well among normal inflammatory conditions, researchers really had to act like detectives here. You see, current diagnoses rely heavily on looking at non-specific features under a microscope. We're talking about things like bands-like dermal infiltrates that honestly could just be a bad case of eczema. It's totally ambiguous. So the team set out to use the raw clinical samples of 18 primary patients as hard evidence to find reliable, indisputable diagnostic hallmarks at the single-cell level.
Moving right along to Section 2, preparing the 18 patient samples. From the clinic straight into the wet lab.
Modern sequencing is just unbelievably versatile. For this study, time was completely of the essence to capture live cells. They took 8 fresh skin samples right from the clinic and ran them through the 10x Chromium platform. But they didn't stop there. They also tapped into 10 formalin-fixed archival tissues, the kind saved for years in paraffin wax blocks, using a special 10x Flex Pro-based profiling method. This dual approach of combining fresh, live cells with historical fixed samples gave us an incredibly rich and robust dataset to work with.
For those 8 fresh samples, it's basically like taking apart a building brick by brick. First, a dispase 2 enzyme is used to literally peel the top layer of skin, the epidermis, right off the deeper dermis. Then, they use a collagenase incubation to break it down even further into a sort of cellular soup. Finally, using flow cytometry or FACS sorting, they sifted through the soup to grab the exact immune cells they wanted to sequence, zeroing in on specific protein markers like CD45 and CD8A.
All right, Section 3, the data processing pipeline. We're transitioning out of the wet lab and into the digital dry lab.
Think of this part as a massive digital filtration system. This rigid sequence of code filtered out all the noise, ambient junk, and random errors from the sequencing machines. First, Star Solo aligned the raw data directly to the human genome. Then, Cellbender acted like a vacuum, sucking up ambient RNA contamination, basically just the loose genetic dust floating around the sample. Next, Scrublet was deployed to find and toss out doublets, which are just 2 cells that accidentally got stuck together and sequenced as one. Finally, ScanPy grouped all this pristine data together. Every single high-quality cell in the final atlas had to survive this rigorous computational gauntlet.
Now, to put this into perspective, the scale of this refinement is just staggering. After running those 18 patient samples through the physical wet lab digestion and then through that intense bioinformatic filtering, the pipeline yielded approximately 420,000 pristine single cells. That's a massive amount of data. And all of these cells were successfully captured, quality checked, and neatly categorized into 11 broad cell types.
Section 4, outcomes, the cellular atlas. Let's actually map out this tumor microenvironment.
Okay, what you're looking at here is a UMAP, which is a way to visualize all that complex data. Our computational pipeline successfully clustered those hundreds of thousands of cells into distinct, colorful little islands. Each tiny dot you see is one of those 420,000 individual cells. Because of that sophisticated processing pipeline we just talked about, the algorithms could easily group cells with similar genetic instructions together. This effectively isolated the bad guys, the malignant T helper 2 cells, from the surrounding benign immune cells.
And this is where it gets really cool. It's a perfect macro to micro zoom.
By placing the actual physical histology of the skin layers right next to our computational clusters, researchers ran spatial transcriptomics, which basically paints the gene expression directly onto a map of the tissue. They could validate exactly where these malignant cells were hiding in the skin. It proves for sure that the computational data isn't just abstract math. It perfectly reflects the biological reality going on inside the patient's skin.
Finally, Section 5, Interpreting the B-cell link. Let's reevaluate what's actually happening in this tumor microenvironment.
Here is the massive plot twist of the study. The transcriptomics revealed a shocking enrichment of B cells grouping around the tumors. So why is this a big deal? Well, normally B cells are absolutely not present in the skin. Not in healthy skin, and definitely not in the benign conditions that CTCL imitates, like your standard eczema or psoriasis. But in these specific cancer samples, the tumor cells were physically associating with B cells, essentially remodeling their microenvironment to create specialized, supportive little structures right there in the tissue.
Grounding all this computational data back into human reality, we see that a higher B-cell abundance directly correlates with disease progression. Notice how the red line, which represents the patients with a high abundance of B cells, shows a much faster drop in progression-free survival compared to the gray line representing low abundance. The data pipeline didn't just map cells, it found a hidden biomarker that predicts a worse outcome for the patients.
So as we wrap up this explainer, we have to ask ourselves an exciting question. How might leveraging these transcriptomic pipelines unlock completely new immunotherapies? By rigorously taking those 18 patient samples from the clinic, digesting them in the wet lab, and translating them through advanced bioinformatic code, researchers illuminated the darkest corners of this disease. If B cells are literally helping these tumors survive, targeting those specific aggregates might just be the key to finally cornering and curing the great imitator. Thank you so much for joining us and learning with us today.